Welcome back to the Deep Dive. Today, we're jumping into something that really challenges, well, a lot of what we think we know
Hmm.
about the global marine environment. We're
hmm
talking marine biological invasions, but maybe more importantly, the system we use to actually track them.
Yeah.
It seems pretty flawed.
Yeah. Deeply flawed is probably the right way to put it. We're focusing on this key paper, The Assessment of Marine Bioinvasion Diversity in History. It's by James Carlton and Evangelina Schwindt, just out late 2023.
Okay.
And it basically argues that trying to compare invasion lists, region by region, it's almost impossible right now. The methods are just too different.
Right, so that's our mission for this dive then, to
Yep.
really get into why these methods are so inconsistent.
Exactly, because the big question is, are these differences in numbers, say, between the Med and Peru, real ecological differences?
Like one place is just better at resisting invasion.
Right. Or is it just down to how scientists looked? What they looked for, where they looked, how far back they went.
And if it's the latter.
Then our whole understanding of marine ecology, invasion patterns, management, it's all built on shaky ground. The baseline's just
Yeah.
off.
Okay, so let's dig into that. This inconsistency, this scientific blind spot. The paper says studies are failing in, what, 3 main areas?
Yeah, 3 big ones where the scope is often just too narrow. It comes down to maybe historical convenience or focus.
Okay, what was the first one?
The first is taxonomic breadth. Basically, for a long time, researchers focused on the easy stuff, bigger things like fish, mollusks, crabs, things you can see easily. And they tended to skip over what the paper calls the understudied taxonomic groups.
Right, the less obvious ones, like what?
We're talking about tiny or soft-bodied things. Think ribbon worms, nemerteans, sponges, hydroids, those segmented marine worms called polychaetae.
Things that need a real specialist to identify properly.
Exactly. If you don't have that expert looking specifically for, say, invasive flatworms, they don't get recorded. They're invisible in the data.
Makes sense. Especially for older studies, maybe focused on fisheries or something.
Precisely. Then the second gap is habitat breadth.
Okay.
Traditionally, marine surveys kind of stop right at the high tide line, but the paper argues, look, that's not enough.
Why not?
Because tons of invasive species actually thrive in these coastal zones that need salt water but aren't strictly underwater all the time.
Ah, like the splash zone or marshes.
Yeah, exactly. They talk about the supralittoral zone, that's the strand line where seaweed washes up, and maritime sand dunes, salt marshes. Organisms there need salt. They interact with the marine system. They should count.
And didn't they even push it to include things like rats?
Yeah.
How does a Norway rat fit into a marine invasion list?
It sounds odd, right?
Yeah.
But think about their ecological role. If an introduced rat is living on the coast and its main food source becomes, say, introduced marsh crabs or shorebird eggs,
Oh, okay.
it's acting as a major non-native predator within that coastal marine food web. It's impacting the structure just as much as an invasive crab might. So they argue, yeah, it belongs in the assessment.
I see the logic there. Okay, so we've got overlooked species, overlooked habitats. What's the third gap?
Historical depth. This one's about time, about the chronology.
How so?
Most invasion lists just use the date of the publication that first reported the species as the first detection date.
Okay. Seems reasonable.
But it's often a really poor substitute. What if the specimen was collected, sat in a museum for 50 years, and then someone wrote the paper?
Right. That completely messes up the timeline.
Totally. It skews our whole perception of how fast invasions happen, and crucially, it makes it almost impossible to link that introduction to why it happened. Like connecting it to a specific shipping route opening or a new canal. Using the publication date breaks that link.
Okay, this is starting to sound like a massive undercount across the board. Does the paper actually quantify this? Like, what happens when you do apply these more rigorous methods?
Yeah, and the results they show are, well, they're pretty staggering. It suggests the existing lists aren't just a bit off, they're wildly underestimating the scale.
Give me an example.
Okay, take Hawaii. The standard lists showed about 90 recognized marine non-native species.
Right.
Using Carlton and Schwindt's comprehensive approach across all the taxa habitats, digging into the history, that number jumped to 333.
Wow. From 90 to 333. That's huge. Almost 4 times as many.
Yeah, a nearly 270% increase. It basically means most comparisons using the old Hawaiian data are just not reliable.
At other places.
Even more dramatic changes in some less studied areas. The Galapagos went from just 5 recorded invasions to 53.
10 times more.
Yep. And down in Argentina, Uruguay, the count went from 29 up to 129.
Okay, so this isn't just tweaking the numbers. This is fundamentally changing the picture of marine invasions globally.
It really is. It shows just how much we've likely missed by not looking systematically enough.
So the paper isn't just pointing out the problem, it's offering a solution, right? A kind of guide to doing this properly.
Right.
What's step one in their rigorous approach? They called it the sine qua non.
That's right. The essential thing you can't do without. And for them, it's establishing a robust and reliable taxonomic foundation. Basically knowing exactly what species you're dealing with.
Seems fundamental.
It is, but here's where it gets interesting. They immediately warn against relying only on modern molecular tools like DNA sequencing.
Really? But isn't DNA supposed to be the gold standard now? You mentioned a molecular paradox.
Exactly. Tools like eDNA sampling or checking sequences in databases like GenBank are incredibly powerful, no doubt. But the paradox is their accuracy depends entirely on the accuracy of the original identification linked to that sequence in the database.
Meaning the initial sample might have been misidentified.
Precisely. Someone, maybe not even a specialist in that group, uploads a sequence with an ID, then later someone gets a 99 or 100% match to that sequence and assumes their sample is the same thing.
But if that first ID was wrong?
The error just propagates. The paper points out studies finding, for instance, that up to a third of the sequences for the common green seaweed ulva in GenBank might be misidentified.
A third. Wow. That could lead to some serious confusion in ecological studies.
Absolutely. And they give a concrete example. A study reported finding an Asian barnacle, Megabalanus rosa, in California based on genetic matching.
Okay, that sounds like a significant new invasion record.
It would be, except when taxonomists later looked closely at the physical specimens, using traditional morphology.
Let me guess. It wasn't the Asian barnacle.
Nope. It turned out to be the native California barnacle, Megabalanus californicus. The reference sequence in the database that it matched was simply wrong.
So the takeaway is, molecules are great, but you still need the experts looking at the actual organisms.
You absolutely need morphology to ground truth the molecular data. They have to work together.
Okay, so once you have your taxonomy sorted, what's next for finding these overlooked invasions? You mentioned literature surveys.
Right. The paper stresses that researchers need to cast a much wider net when searching the literature. You can't just search for modern terms like invasive or non-indigenous.
Because older papers use different words.
Exactly. You need to include older terms like tramp species, adventive, colonist, even xenodiversity. These were common in the 1800s, early 1900s.
So it's almost like needing a historical linguist on the team, too.
Or at least being aware of that historical terminology. And crucially, they say, you have to look beyond just regional ecological journals. You need to hunt for what they call hidden invasion records buried in global taxonomic monographs or expedition reports.
Can you give an example of that? A hidden record?
Sure. The European salt marsh snail, Myasotella myosotis, it was present and established in South Africa for over 100 years before anyone in the South African marine science community realized it was non-native.
A century. How was it missed?
Because the record confirming its presence there wasn't in a South African journal. It was tucked away in a global taxonomic revision of related mollusks published elsewhere. Unless you were digging deep into that specific taxonomic literature, you'd completely miss it.
Wow. That really highlights the detective work needed.
It absolutely does. And this deep historical digging also helps challenge another big problem: the shifting baseline assumption.
Okay, what's that?
It's the tendency to assume that any species found and recorded really early on, say in the 1700s or 1800s during initial biological surveys, must be native to that place.
Just because it was found early?
Pretty much. But humans have been moving species around for centuries, even millennia. So this assumption means lots of very old introductions get mistakenly labeled as native. The paper calls these pseudo-indigenous species.
Pseudo-indigenous.
Uh-huh.
So basically long-term invaders hiding in plain sight accepted as local fauna.
Exactly. And they've been potentially distorting our understanding of the native ecosystem for a very long time.
So how do you actually challenge that status? How do you unmask a pseudo-indigenous species after centuries?
The paper lays out 14 different criteria, like evidence types, to determine if a species is non-native. One really powerful one is criterion 12, association with other non-native species.
How does being associated with another invader tell you anything?
Well, think about it. If you find, say, a parasite or a commensal organism that lives only on a known introduced species,
Ah, it probably hitched a ride with its host.
precisely, or if you find a predator that only eats a known introduced prey species in that region, the logical inference is that the associate, the parasite, the predator, is likely non-native too, even if it was recorded there earlier and thought to be native.
That makes sense. It's like guilt by association, but ecologically sound.
Is there a specific case study for this?
Yeah, they mention a predatory flatworm, Stylus planolimnoria. It was first described from Los Angeles Harbor, so initially maybe assumed native to California.
Okay.
But the key thing is, it's only ever been found preying on one specific thing,
Mm.
the introduced wood-boring isopod, Limnaria tripunctata.
So its entire food source in that area is an invader.
Right. Since its lifestyle is completely tied to that known invader, the conclusion is the flatworm itself must also be an introduction. It likely arrived with its food source.
That's a clever way to connect the dots historically, linking it to the vector that brought the prey.
And speaking of history, let's circle back to the timing issue. We know publication dates are unreliable. How does this deep dive approach fix that?
It means doing the archival work. Instead of grabbing the publication date, researchers need to track down the actual collection date.
How? Where do you find that?
You have to go to museum records, look at specimen labels, check old expedition logs or station data sheets. It's real historical detective work.
And does this significantly change the dates?
Oh, absolutely. It can completely rewrite the invasion timeline for a region. They give the example of an ascidian, a sea squirt called Polyandrocarpa zoratensis in Peru.
Okay.
The literature said its first record was from a 1931 publication.
Right.
But when researchers tracked down the original type specimen in the Yale Peabody Museum,
It's older.
much older, the collection label showed it was actually collected way back in 1866.
Wow, 1866 versus 1931. That's a 65-year difference.
Exactly, but it doesn't just change a date, it changes the whole context. Suddenly, you're looking for potential vectors like early steamships or specific trade routes active in the 1860s, not the 1930s. It connects the invasion to a completely different historical period.
Okay, so this rigorous approach, checking taxonomy, habitats, history, associations, it paints a very different picture. Why is getting this right so important for, say, policy or management?
Well, it's crucial for a few reasons. First, it helps managers distinguish between species that arrived via human, help the ones policies usually target, and species that might be expanding their range naturally, perhaps due to climate change.
Right, because you'd manage those differently.
Completely differently. Although, interestingly, the paper's definition of bioinvasion is broad, any species establishing via any vector, human or natural. But policy usually focuses just on the human-mediated ones.
Okay, what else?
It fundamentally challenges our view of global invasion patterns. You know those heat maps you sometimes see, showing where invasions are supposedly worst?
Yeah, often North America, Europe, Australia look like hotspots.
Exactly. But the paper warns, those maps might be showing us less about where invasions are actually most intense, and more about where scientists have looked most intensely.
So they reflect research effort,
Yeah.
not necessarily biological reality.
To a large extent, yes. Regions with more funding, more taxonomists, more historical records available, like Europe and North America, inevitably report more invasions.
Which could mean areas like the tropics, often shown with fewer invasions, aren't necessarily less invaded.
They might just be drastically understudied. The data just isn't there because the effort hasn't been applied consistently.
Do they have an example of that potential undersampling?
They mentioned Singapore. A detailed review there listed only 22 confirmed non-native species. Seems low for a major global port, right?
It's surprisingly low.
But that same review also noted that for crabs alone, over 127 species were listed as cryptogenic,
Meaning?
meaning their origin is unknown. They couldn't be confirmed as either native or non-native.
So potentially, many of those 127 crabs could actually be introductions.
Potentially, yes. And that's just crabs. The review noted a near total lack of records for other major groups like sponges, hydroids, ascidians in that region. It suggests a massive blind spot, likely typical of many tropical areas. The absence of evidence isn't evidence of absence.
So bringing it all together, the paper's main message seems to be, We really don't know nearly as much as we thought about the true scale of marine invasions.
That's a big part of it. They argue that without this kind of thorough, vetted, historically deep assessment, our ability to understand marine ecology, evolution, and definitely our ability to create effective management and policy is seriously compromised.
We're operating with incomplete, potentially misleading data.
Exactly. Their core advice to scientists and managers is pretty blunt. We need to stop making strong judgments about where invasions have or haven't happened based on the current inconsistent data. We need to acknowledge the huge gaps.
So for you listening, what's the key takeaway here?
I think it's that when you see a map or read a headline talking about numbers of invasive species in a particular place, take it with a grain of salt.
Remember that number likely reflects, to a large degree, where scientists have looked and how they've looked. The actual true number of species moved around by humans living in places they didn't evolve in. It's probably far, far higher, and the process started much earlier than most current estimates suggest. A final thought, then, something to chew on. If just applying these rigorous methods can take the count in a place like the Galapagos, from 5 known invasions to 53, just imagine how many species currently listed as native all around the world might actually be these pseudo-indigenous invaders introduced centuries ago, just hiding in plain sight.
It's a staggering thought, isn't it? Thousands potentially, still waiting for their true history to be uncovered.